everyone! Today, I'm going to talk about a technique that's super important in cell culture, cryopreservation. It's a crucial process that helps preserve cells, tissues, and even entire organisms at very low temperatures, and it's especially useful in research and clinical applications. So, what exactly is cryopreservation? Well, it's the process of freezing biological samples, like cells, stem cells, sperm, or embryos, using liquid nitrogen to stop all biological activity. This prevents the cells from degrading over time. But it's not as simple as just freezing everything. If done wrong, it can actually cause damage to the cells, which is why it's so important to get it right. Cryopreservation is essential for storing biological materials long-term. For example, if we want to keep stem cells or sperm for future use, cryopreservation helps ensure they stay viable when we need them. It's especially key in places like fertility treatments or biobanking, where we need to preserve the quality of the cells for later. Now, let's talk about how to actually do it the right way. First up, we need to prepare the sample properly. You may not know this, but most biological materials are made up of water, and when water freezes, it expands. If cells freeze without the right precautions, ice crystals can form inside and damage the cell membrane. So, to prevent this, we use a freezing medium, things like dimethyl sulfoxide, that helps protect the cells during the freezing process. It's important to get the concentration of the freezing medium just right. Too much can actually be toxic to the cells, so it's crucial to find the right balance. If you're unsure, don't hesitate to reach out. We're happy to provide free technical support to guide you through it. Once the cells are prepared, we need to freeze them gradually. Freezing too quickly can cause uneven cooling, which could damage the cells. Ideally, we use a controlled rate freezer, which cools the cells slowly and evenly. If a freezing container is not available, you can manually freeze the cells by sequentially placing the cryovials at 2 to 8 degrees Celsius for about 40 minutes, then at minus 20 degrees Celsius for 30 to 60 minutes, and finally at minus 80 degrees Celsius overnight. After the cells are frozen, they're stored in liquid nitrogen at minus 196 degrees Celsius. This super low temperature essentially stops all biological activity, keeping the cells preserved until we need them again. But freezing is only half the battle. Thawing is just as important. If you don't thaw the cells carefully, you could end up causing even more damage. The key is to thaw the cells rapidly, usually in a water bath at 37 degrees Celsius. This helps prevent ice crystals from forming again. You also need to handle the sample gently, as any rough handling can harm the cells. Once the cells are thawed, it's time to remove the freezing medium. This is done gradually to avoid exposing the cells to too much cryoprotectant, which can be harmful as they recover. So, to sum it up, cryopreservation requires careful preparation with the right freezing medium, gradual freezing, and gentle thawing. When done properly, it preserves the cells for future use. But even small mistakes in the process can lead to damage, so it's really important to follow the steps carefully. If you found this video helpful, make sure to like and follow for more tips on cell culture. Thanks for watching.